I am happy to be here, and in the following approximately half an hour, I will be happy to share with you our experience with what happens in the porcine intestinal tract, how porcine gut microbiota develops along the whole life of pigs, what happens in gestating sows, in piglets, weaners, as well as in pigs, fattening pigs in commercial production. Obviously, or currently, we are looking at the composition of gut microbiota, but overall aim and goal of our activities is to identify bacterial isolates with positive effect on pig health, and as early in the future as possible to use such bacterial isolates as probiotics. Now, you may immediately argue with me that you have heard a lot of different presentations on gut microbiota, and the reason why gut microbiota or whatever microbiota, whatever different microbial communities and studies on these topics became quite popular is given in this slide. Approximately 10 years ago, new platforms for DNA sequencing, nucleic acid sequencing, have appeared on the market. And these technologies are based on purification of total DNA from a sample of your interest, fragmenting of these original whole chromosomes into small pieces of DNA, small fragments, and extensive sequencing of each individual fragment, let's say one by one. Current platforms, current instruments can very easily read 15 million randomly generated DNA fragments overnight. And if you have 15 million sequence information, you can then submit these sequences into appropriate databases, and you can extract from the database information that sequence of this green bacterium is similar or equal to E. coli. Sequence of this red fragment is similar or equal to Bacteroides. Sequence of this purple fragment is the same as has been deposited for Mycoplasma. And counting how many fragments out of these 15 million belong to each individual bacterial species allows you then to trace back the composition of the material which you have tested, which you are testing. So you do not need culture, you sequence nucleic acids and you get an information on the composition of microbiota in your samples which you are analyzing. This is exactly the technology which we are applying for studying of gut microbiota in farm animals. Today I will be talking about pigs. My presentation will consist on results of 4 different studies which we have performed in pigs so far, and how different conditions, different experimental interventions affect composition of gut microbiota in pigs. The first study which we performed was done in collaboration with Italian, French, Danish and Swedish partners. And first we asked and questioned ourselves whether microbiota composition in pigs originating from conventional herds or from organic herds, whether this different production system will influence composition of gut microbiota. We expected that we expected that there would be differences, because pigs in the organic farming conditions, under organic farming conditions, are exposed to much greater selection pressure from the environment. And in this particular study, we also addressed distribution of antibiotic resistance bacteria in microbiota of pigs kept under these 2 conditions. In that case, we expected that lower abundance of antibiotic resistance will be recorded in organic pigs in comparison to pigs kept under conventional conditions, since antibiotic usage is highly controlled and restricted in organic farming. Now, In this slide, I immediately show you the results. Results on the composition of gut microbiota. I will be commonly using these figures in the rest throughout of my presentation. And in these figures, principal component analysis, or principal coordinate analysis in this case, this is the statistics in behind. And in these figures, every individual spot represents one particular pig. In our case, pig and his fecal material and microbiota determined for this particular pig. If two spots are close to each other, this means that these two pigs exhibited or harbored similar or even identical microbiota. And these two pigs, in comparison with, for example, these two, they harbored relatively different microbiota. Now, if you follow the colors with me, Italian pigs, or samples originating from Italian pigs, French pigs in red, Swedish and Danish in green and blue, you can immediately follow with me and spot with me, identify with me that red and yellow spots are a little bit separated from those in green and blue color. So microbiota in pigs originating, or in fecal material originating from pigs from southern countries, Italy and France, a little bit differed from microbiota present in pigs originating from Sweden or Denmark. Geographical distribution was relatively likely, but if you now focus on full symbols or empty symbols, identifying pigs originating from conventional farming systems or organic, you will probably did not identify any difference. These different spots, open and filled, they overlap each other, what means that being grown under conventional conditions or organic farming, unlike our expectations, did not influence composition of gut microbiota nearly at all. No importance. This figure shows you nearly the same as the previous one, but every individual pig is identified along the x-axis, and y-axis tells you total microbiota composition in pig number 1, originating from Italy and organic farming conditions, and so on. Even if you just follow the colors with me, you will see that there is a nice total mess. No real profile, nothing what would distinguish one group of pigs from the others, and these are organized according to Italian from organic farming, French organic, Danish organic, and so on. You may perhaps identify minor differences in here. Light blue color is present in majority of these individual pigs in here. Light blue color is relatively loudly represented in pigs originating from conventional production systems in Denmark and Sweden. Light blue means representation of Prevotelaceae. One particular group of bacteria is loudly represented in Swedish and Danish conventional pigs. This bacterium is highly present in all the remaining. This a little bit explains the differences which I highlighted in this figure. Danish and Italian and French are different from Swedish and Danish. Now, in this study, we also look at the distribution of antibiotic-resistant genes. In this case, we used quantitative real-time PCR, and we determined the abundance, incidence, presence of selected genes which are known to be responsible for antibiotic resistance. And in this case, this is STRA gene, resistance to streptomycin. Now, each box here indicates whether the samples originated from Italy, France, Sweden, or Denmark. Black box, incidence of antibiotic resistance genes in pigs originating from conventional farming systems. Green boxes, incidence of antibiotic resistance in pigs, or in microbiota of pigs originating from organic farming. If you then compare within the country, comparison boxes, which are always next to each other, conventional, organic, no significant difference in Italy. Conventional organic in France, no significant difference. Conventional and organic in France, no significant difference in the distribution of streptomycin resistance among gut microbiota in pigs. But what you can immediately spot, drop in between all French or Italian samples in comparison to all Swedish or Danish for STRA gene, for SOL1 gene, sulfonamide resistances, higher incidence, higher abundance in Italian and French, lower incidence, significantly lower incidence in Swedish and Danish. Lesson taken from this, being rather surprising, being organic or conventional, although you use or do not use antibiotics more or less frequently, doesn't affect that much, too extremely, the presence of antibiotic-resistant bacteria in the fecal microbiota of your pigs. More important is the geographical location, whether you are located in south of Europe or north of Europe. And of course, I'm not thinking of geographical location itself in terms of geography, but rather climate conditions, epidemiological conditions, and the most likely really countries which are down to the south, countries where outdoor temperatures during summertime approaches 35 degrees, 37 degrees, those temperatures which we use in incubators for bacteria propagation. It is simply more difficult to keep farm animals healthy without the use of antibiotics. They are simply forced to use antibiotics due to the external conditions, and this is reflected in the figure here. This is valid for streptomycin resistance, sulfonamide, another variant of sulfonamide resistance. This is also valid for tetracycline resistance, here and here, two different genes. So relatively clear message from me. This is the same figure, or showing the same data as the previous one, but in a different way. Principal component analysis. Each spot again tells you positions in each individual pig. Now the distribution is based on the distribution of genes responsible for antibiotic resistance. Swedish and Danish pigs are clearly separated from those originating from Italy and France. Perhaps in Italy it is the situation a little bit different than in French pigs. But whether closed symbols or open symbols, whether the pigs originated from organic farming or conventional farming, did not affect the incidence or abundance of frequency of antibiotic-resistant genes in bacterial populations forming gut microbiota of these pigs did not influence this too much. Now in next study, so I'm switching to another experiment, another study. This experiment has been mentioned yesterday in one of the previous talks. In this study we addressed whether housing conditions of sows affects composition of their gut microbiota, and the sows during the gestation were kept either under conventional conditions on a slatted floor or enriched conditions, and the main difference was square meters per sow, and in terms of, or in association with composition of gut microbiota, presence of straw bedding on the floor in the case of enriched conditions. Once or after farrowing, sows with their piglets from both cases were kept under the same conditions. And we then tested differences in gut microbiota of sows originating from these two conditions, and we then tested also gut microbiota composition in their piglets. We found out, a little bit as expected, that sow microbiota of all the sows, clearly different from microbiota which we identified in one-day-old piglets, and microbiota from one-day-old piglets clearly differed from microbiota which we identified and characterized in four-day-old piglets. In the piglets, now looking at the small symbols or big symbols, there were no real differences in microbiota composition, whether the piglet originated from the sow which was kept under enriched conditions or conventional. Keeping the sows and under different conditions didn't influence composition in their piglets. But within the sows, big symbols are a little bit clustered toward the top of y-axis. Lower symbols, smaller symbols, conventional sows, a little bit moved down to this. So there were minor differences among the microbiota or in microbiota of sows kept under these two different conditions. If you look at specifically what bacteria were present in sows and their piglets, the fact that they differed clearly on a previous slide, as I showed on the previous slide, it can be seen in this slide as well. Microbiota of sows was dominated by number 5, green color Prevotella, but followed by Oscillospira, followed by number 8, Luminococcus. Transfer of sow microbiota to their piglets was nearly zero, was absent, or was absent. It was in there. I do believe that the transfer was present. But since the feed was completely different, milk in the case of piglets and rich full feed composition in sows, the feeding regime, milk, selected for completely different bacteria in the young piglets, doesn't matter whether 1 day old or 4 day old, and these bacteria clearly differed from the bacteria which were dominant in the sows. When we specifically looked at the microbiota of the sows for the differences in abundance of individual microbiota members, we found out that microbiota of conventional sows or sows kept under conventional conditions was significantly enriched, for example, for lactobacilli, for Bouledia, Lactospira dorea, Luminococcus oscillospira. If you asked me before, I would say that all these bacteria are usually indicators of healthy microbiota. On the other hand, sows kept under enriched conditions were enriched for Prevotella, brownish columns, Parabacteroides, and other 2 species which are still awaiting for their names, and Fibrobacter. Very likely, all these bacterial genera, are known to be adapted to fiber degradation, and very likely their increase has been caused or influenced by the consumption of the straw from the bedding by the sows and enrichment of their diet of the sows in enriched cages, enriched spaces, enriched conditions, positively selected for the presence of bacteria which are capable of degrading different fibers present in the diet. Colors here means that those are gram-negative bacteria. Their terminal products of their metabolism is usually acetate and propionate. These bacteria belong to gram-positives, and their terminal products of their metabolism is usually acetate and butyrate. Now, third study, which we performed in collaboration with French partners. You have heard about experiments with low residual feed intake pigs and high residual feed intake pigs yesterday. So these pigs were monitored not only for different biochemical markers and behavior changes, but also for the composition of gut microbiota. In the first experiment, we tested or we compared development of gut microbiota in weaned piglets. So this is the scheme of the experiment. V stands for the week. So we started with 4-week-old piglets, which were just weaned, and we followed development of gut microbiota in the piglets belonging to these 2 genetically different pig lines. What we found out, age dominated over the influence of genetic background. Piglets aged 4 weeks form a separate cluster from the remaining piglets or microbiota, And you can hardly see any or minor differences in the composition of that microbiota in the older piglets, but there is some kind of difference in between of the piglets originating from low residual feed intake line or high residual feed intake line, especially at the time of weaning. When we looked specifically at the individual microbiota members which might be responsible for these differences, we identified that Bacteroides and Clostridium 14a isolates were enriched in weaners, in weaned piglets belonging to low residual feed intake line, and other bacteria like Prevotella, Megasphaera, Putitri-Vibrio, or Fecalibacterium were significantly enriched in the microbiota of piglets belonging to the high residual feed intake line. Now I will now go for study number 4, and then I will combine everything into a single final slide. In study number 4, we tested development of that microbiota in the same pig lines, but the age was different, pigs were older, and as mentioned yesterday, half of the pigs in each group were subjected to sanitary stress lasting for 7 weeks. Composition of that microbiota overall is given in this slide, and unlike the weaners, in this case the age-dependent development is already a little bit diffused. You can still see red colors of the youngest animals, youngest pigs on one side, green and blue spots on the other side, older pigs, so there is still some kind of development of that microbiota, but not as extensive as in the youngest piglets. Now, when we wanted to decide whether some bacteria are really associated with either high residual feed intake line or low residual feed intake line, we combined these last two experiments into a single analysis. So we really wanted to say that if particular bacterial genus is really associated with low or high residual feed intake line, it should be differently present in both young weaned piglets as well as in the fattening pigs. And this figure tells you the results. Each individual column here tells you red, low prevalence or abundance in total microbiota of lactobacillus in high residual feed intake line and high residual feed intake line and pigs of the same age, 4-week-old. I can't put numbers in there. 5-week-old, 6, 7, and then start the second experiment with 12, 17, 19, 23, and so on and so on. Just visually compare the distribution of red and subsequent green columns. And you will identify with me, asterisks indicate statistically significantly different abundance, but the overall profile confirms that really Lactobacilli, Fecalibacterium, Megasphaera, and the other bacteria are preferentially associated with pigs belonging to high residual feed line. Nearly always the red columns are higher. If you ask me a similar question to myself, you asked me before, I would identify this bacteria as of positive effect on pig gut health. Majority of Lactobacilli, I don't need to discuss with you, everybody probably accepts Lactobacilli as markers of positive gut health. Fecalibacterium, Megasphaera, prototype butyrate product producers, Butyri-Vibrio and other butyrate producers, Olzenella and Collinsella, these are similar to Bifidobacterium and belong to Actinobacteria. So from the composition of gut microbiota, high residual feed intake pigs and piglets somehow represent those which are populated by a healthier microbiota. Now, let us look at the composition of gut microbiota in low residual feed intake pigs. Which bacterial species were enriched in this line? I would stress mainly Oscillibacter and Bacteroides. It seems to me, and our conclusion was, Bacteroides. As I have already mentioned, these isolates or strains belonging to genus Bacteroides, they are usually capable of fiber degradation. So very likely, low residual feed intake pigs are capable of more efficient fiber digestion, something which is not available to high residual feed intake pigs because they have lower abundance of Bacteroides. And I also noticed and specifically looked at the Oscillibacter because species name of Oscillibacter is Oscillibacter valerizigenes. It ends up with production of valerate, C5, 5-carbon fatty acid. Whether this fatty acid, whether Bacteroides digests fibers, produces acetate, and this acetate is uptaken by Oscillibacter and converted into valerate, which might be then consumed by pig host as a fatty acid, high energy source. This remains to be determined, but this should or could explain why these bacterial species are more abundant in low residual feed intake pigs, and this could explain why low residual feed intake pigs can extract more energy from the feed provided than the high residual feed intake pigs.
Ivan, 3 minutes.
Okay. When we looked at the influence of sanitary stress, relatively minor changes. We identify only 4 bacterial genera which significantly changed in the abundance after exposure to sanitary stress, which means quite a few. However, I would remind that one of them was Prevotella, and Prevotella is one of the most important, most prototype, most abundant bacterial species present in or forming porcine gut microbiota. And if you change abundance of the most important, most abundant microbiota member from 40 to 30% or even to 20%, this may indicate that although number of species which have changed after sanitary stress is low, importance might be relatively moderate or relatively high. Now, my last scientific slide with some kind of content. Finally, we combined all of our studies into a single figure, and this is that one, summarizing what happens along the whole life of pigs. During the first day, really immediately after birth, gut microbiota is dominated by Enterobacteriaceae, by E. coli. Soon after, E. coli and Enterobacteriaceae decreases the abundance by something like 1 or 2%. Fusobacteria appear, and immediately after, gram-positive representatives of Firmicutes, different shades of green, appear, and as well as gram-negative bacteria from the family Bacteroideaceae. At the time of weaning, nothing dramatic happens here, but Bacteroideaceae and Bacteroides is replaced by Prevotella and Prevotellaceae. Why this is so, we do not know at all, but definitely lactation and complex feed is responsible for this switch. If I am going to summarize what I wanted to share with you, what I wanted to tell you. So, just the previous slides, development of porcine gut microbiota can be divided in 3 different periods. In the very first days of life, Enterobacteriaceae dominate. During lactation, except for the day 1, Bacteroides and Bacteroideaceae are characteristic for the normal gut microbiota, but immediately after weaning, Bacteroides is replaced by Prevotella. Reasons relatively unknown. If you go for environmental factors like organic farming or sanitary stress, rather counterintuitive, these are of a lower importance. However, mind, not really the direct environmental factors, but geographical location. This may influence gut microbiota composition, and in particular, it really influences distribution of antibiotic resistance among gut microbiota. There is clear direct correlation, positive correlation going south, higher abundance of antibiotic resistance. Feed composition, and now I mean extreme comparison, lactation, milk, and standard feed. This influence composition of gut microbiota quite a lot, and in fact this influence dominates over the contact with adult sow. So selection pressure, investment of gut microbiota in their piglets in the very first days of life is nothing against the effect of milk itself. And finally, genetics. Genetics indeed affects composition of gut microbiota, although at the present I have mixed feelings. High residual feed intake pigs, for the first point of view, I would immediately say enriched for microbiota members which are considered as healthy. Low residual feed intake pigs, which probably would be of more interest to most of you, most of the producers, because you feed them less to reach the same weight. Enriched for bacteria which intuitively are not associated with a positive gut health, but definitely they are performing better in terms of feed conversion, feed utilization than the high residual feed intake pigs. Future experiments focus really on what happens here. Within 1 week, the most important bacteria, or two, the most important microbiota members invert. Invert in such a way that Prevotella forms something like 5% only, Bacteroides forms something like 25% of total microbiota, and within 7 days after weaning, the ratio completely exchanges, and different changes or changes in gut microbiota immediately after weaning is dramatic or extensive also in other bacterial species forming gut microbiota. Now really, all from my point of view, I would like to acknowledge the contribution of different partners across the whole Europe, from Italy, France, Denmark and Sweden. Current collaboration is under the ProHealth project with colleagues from India, France. And I also have to thank all my collaborators in my group who in fact did all this work. I am only presenting out.